Well, I welcome you all for this session on tools to access and interact with publicly available data. As you know, more and more organizations are starting to follow open data initiatives, so that provides us access to a lot of public data. So the access to a lot of data also presents us with lots of challenges that have to do with integration of data, finding common themes across data sets, various ways to integrate data sets, and things of that nature. As you can imagine, if you're talking about open data access, these are the data sets that are coming from various organizations. And usually these data sets are generated in silos. When data is generated in silos, it poses a lot of challenges in integration. And if forget about integration of data sets across organizations, even if you take a very large organization and various divisions within those organizations, mostly they work in silos. So all the data that is being generated, even within an organization, poses tremendous challenges in analysis and integration. So the fact that we have a set of speakers lined up talking about how to leverage public data sets in order to do a lot of different things is a testimony to the fact that we are making progress in leveraging public data sets in order to create knowledge base. With that, I'm going to invite myself for the first talk. I'm going to describe to you this cool tool called Protein Miner that I have developed to do various things when people are trying to learn about proteins of interest. So one of the things, although this topic could be presented in several different ways, the way I want to present it is what can you do currently using all the public sources and what challenges that you can run into and how easy it is to build a system such as what I'm going to show you in a minute. And how can you tackle those problems, right? So when I proposed this topic, one of the challenges that I took upon myself was that I started just with an idea. So when I proposed this topic, I didn't even have a product, right? So I started with an idea, and between proposal of the idea and it got accepted as a talk, and today we have a product. Right? So that's how fast we were able to build something like this. So I'm going to show you a few databases that we worked on, and then we are expanding it to other databases. But this is just to show you how fast you can build something like this using basic technology. All right. So what is a protein miner? So I want you to imagine that you're working on a hypothesis, right? And then you have certain proteins of interest that you want to look into. And how do you leverage public databases in order to extract information from various systems? So if you can think of systems like GPA, Gene Cards, Bioportal, and Open Targets, they have a unique niche. Open Targets, you're talking about targets of interest corresponding to the proteins. GPA is mostly in the transcriptomics space. Gene Cards iterates through various databases. And then provides you basic information. But none of these is actually talking about integration across various systems. These are all sequential query mechanisms, either individually query independent databases, or they provide you information corresponding to each of the databases without actually integrating them. And most of these are usually single gene, single target, and single disease query databases. So if you provide a lot of genes as input to these systems, they actually give you individual information without actually integrating them, okay? So we took the first challenge, which is to take multi-gene, multi-disease, and target approach in order to integrate and summarize information. And this is not a unique concept, right? You can think of these commercial systems which charge you millions of dollars just to get you access to a user interface that queries essentially the same systems mostly. So how fast can you essentially build a system like this? So we try to build this in 2 sprints, each sprint being 2 weeks, and 1 sprint for user interface. So essentially in a month and a half, we were able to build a system that could do a lot of things that I'll be talking about. So what databases did we focus on? I categorized them into different segments like gene expression, gene-drug relations, protein expression, gene-disease relationships, and so on and so forth. So one of the first things that we did was to actually extract information from each of these databases and provide basic information, which is block number one. And block number two is integration of these data sets in order to do various things. So you're starting off with a hypothesis. You want to know whether that hypothesis is valid. You don't know anything about a particular topic. You're trying to generate a hypothesis. Then you can use the system to generate a hypothesis. Or you're just trying to integrate systems for learning about things. You could integrate these systems. You can have knowledge discovery-based system and so on and so forth. Okay. Okay, so I want to demonstrate the utility of this tool using use cases, right? So I want to start with a very easy, basic use case, which is finding the genes that interact with a protein, GAA, and that enriches the glycogen degradation pathway. So we pretty much know the information here. We know the gene that we're interested in. All we need to find is the proteins that interacts with. We also know the pathway that we're interested in, okay? And if you're using public resources, how challenging is it to do something like this, right? So one of the favorite tools that people can go to is GeneCards, which is essentially a free tool where you can plug in a gene name or gene symbol and give you all the information corresponding to that gene. And one of the things that it does is that it gives you top 5 interactors corresponding to that gene. And that's a good starting point because that's what essentially what we are looking for. And to find enrichment, to do enrichment kind of analysis where you're trying to find if a certain pathway or gene ontology term is not selected by chance, but by statistical significance, you need much more than that. So if you try to expand it, it'll give you a maximum of 25 interactors. That by no means is all the information that's present in there. So even if you were to believe that these 25 interactors would give you something meaningful, we'll try to do that. So I took all these 25 interactors and plugged it into Gene Analytics, which is also a tool that comes as a paid subscription within GeneCards. So you go to GeneCards, under analysis tools, it has Gene Analytics, and you try to use that, it gives you all these pathways. And I don't see a glycogen degradation pathway in there. So it pretty much works as a black box. So where do I go from here? Pretty much a dead end for me. Okay, so there are other ways to do this, right? So I could use a very popular database, which is open source, called StringDB, and then do the same, iteration, provide it with a protein gene symbol and ask it to give me everything that interacts with it, okay? So believe it or not, even StringDB has a limitation on number of proteins that it can provide you as an output. It happens to be 25. And then you take all those interactors and then use another tool called EnrichR, which pretty much queries multiple pathway databases and provides you with information on whether something is enriched or not. And again, it gives you something similar to what I'm looking for, but by no means it gives me the information that I'm exactly looking for, right? And then I used a third system called Pathway Commons, which is very rich. It provides you a mapping between genes and pathways that they correspond to. This is essentially what Protein Miner also uses in the backend. So the challenge now is the user interface that Pathway Commons provides gives you that information. I know for sure that using the data that comes out of Pathway Commons, I'm able to answer this question, right? So I use Pathway Commons, I query the gene GAA, it gives me all the interactors corresponding to GAA, and I can use Pathway Commons again because it specializes in pathway databases. I can ask for all the enriched pathways. It gives me all the enriched pathways, but again, nothing corresponding to glycogen degradation pathway 2. It gives me something corresponding to glycogen, but that's where it ends. But essentially, this provides me with a black box, and I don't know why some things are working and why some things are not working. And then I know using 3 different systems, I'm seeing multiple pathways that don't necessarily overlap with each other. So I developed this system called Protein Miner. Remember, it has all the integration of all the databases that I talked about, which itself was a challenge that we tackled in 2 sprints, and then 1 sprint to create a UI. So it has a very nice UI where you'll be able to ask these questions, and it'll give you answers like this. And I can choose and select what kind of interactions, right? So I selected those interactions that actually have physical interaction. So you could have complex of something that regulates the expression of something else and things of that nature. So you can select what different interactions people are interested in. So in this case, I selected physical interactions. And then I asked a question, what pathways are under? So it gives me this complex network, right? And then if I were to take that network and look at all the pathways, you can see that all the pathways are in hexagons and all the genes are ellipses, right? And then it shows glycogen degradation pathway right here, and I know what genes are part of that pathway, okay? And I also know these physically interact with each other based on the information that I started with. Okay, so I can start here, I can go into enrichment analysis, I can zoom into the pathway that I'm interested in and go from there and zoom into the proteins that fall in as part of that pathway. And then I can look into further evidence as to where this information is coming from. And then I can zoom into those manuscripts that provided that information. So I can take this whole pipeline from end to end, start with information all the way to knowledge discovery. Okay, for use case 2, I want to go into a use case which is a little bit more complicated than we started with. In this case, I'm trying to find microRNAs and genes associated with infantile Pompe. So as you can imagine, there are microRNAs, there are genes, and then a disease, right? So there are 3 different things that I'm looking into. To start with microRNAs, I analyzed the data set that was publicly available, and it provided me with about 16 to 18 microRNAs. I added a couple of more based on the internal assay that we had done for Pompe. Other than taking what popped out of this experiment, I also added a couple of more in there. And then for genes that are interesting in the context of infantile Pompe, I analyzed another data set. And as you can see there, that in comparison to the controls here, the people with the disease before the treatment, they had difference in expression. And after 52 weeks of treatment, the expression is kind of similar to the controls. Suggesting that these people, these genes are interesting because that the expression was reinstated in those individuals. And in comparison, if you look at individuals who did not respond to the treatment, you see the comparison between how the expression looked at 0 weeks in comparison to 52 weeks here. Okay, so there are a lot of genes that are differentially expressed here, 1,660 genes. So one of the things that we wanted to test is, if I were to use a large input kind of like this, does the system scale? Okay, so before I wanted to explore the public databases in order to answer this question, right? So I went to the popular database that's out there, which is MidDB, and I entered the 1 microRNA as input, and it gives me all those genes that it's supposed to regulate, okay? And now I have to do this multiple times. If I have 16 microRNAs, I have to enter these microRNAs one at a time. It'll give me all these associations which I can download. Then I have to filter out based on my differential expression list, and I have to filter out based on other information that I'm interested in, which is the disease, and I try to integrate all of that. And if somebody who's not data savvy, somebody who's a basic scientist, this might be a nightmare. So I try to find something else that people might have thought about this question and probably try to address this question. And I was very happy to find that there was a database called MidNet that tried to integrate multiple systems. So essentially, you could provide it with a list of genes, you could provide it with a list of microRNAs, and you can choose a disease, and it'll provide you all the mapping between them. So the challenge here is that it doesn't necessarily map to the input set that you're giving. For example, you enter 10 genes, it'll find all microRNAs that associate with 10 genes, not necessarily the microRNAs that you provided as input. And the same with microRNAs. It'll give you all genes that are associated with those microRNAs, not necessarily the input that you have provided. So it gives, believe it or not, this is a network it generated. So if I didn't tell you this was a network, you probably wouldn't even know what this is, right? So how do you navigate through a complex network like that, right? So that pretty much is a dead end. And again, it lets you download a data kind of like that. And you have to be, again, somebody who's well-versed with data science based techniques in order to explore and extract information from those files. Okay. Protein miner, I could limit all my queries to the proteins that I'm interested in, the microRNAs that I'm interested in, and the disease that I'm interested in, okay? So it generated this nice network where I have the yellow diamonds, which are all glycogen storage diseases. Which is essentially infantile pompa is one of the glycogen storage diseases, and then I have these ellipses which sort of give you the genes that are interacting with these glycogen storage diseases, and I can color them based on their expression. Remember, I started with the gene expression data, so there are genes which are highly expressed in control in comparison to the disease, and there are genes that are highly expressed in disease in comparison to the controls. So I can color them either red or green based on which spectrum they fall into. And then I have these purple hexagons, which are microRNAs. And now that gives me with an opportunity to zoom into this network and focus on the disease that I'm interested in. So again, I'm picking glycogen storage disease. The glycogen storage disease, and then all the network corresponding to that. So out of that complex network that I showed you here, I can zoom into the network that I'm interested in, and I know what microRNAs are relevant to that disease, which happen to be MIR3P, 13P, and 3P, and the second microRNA. I can't read it because of the font here. So anyway, it gives me all other diseases, which sort of suggest the comorbidities associated with Pompe disease. And then you have this expression data that you can overlay on top of it, red being highly expressed in disease in comparison to the control, and green being highly expressed in controls. And then you can start asking other questions. So one of the questions that we asked is, given MIR 13P, 133A, and 203B, what common biological processes are associated with this microRNAs of interest? So you can start asking this question. So one of the ways you can ask this question is to take all those proteins that are associated with these microRNAs and find the enriched biological processes. And I'm showing you a snippet of all the biological processes that are associated with these microRNAs. So you can go a roundabout way in order to ask this question. And one of the ways that the reason we did this is because there are currently no databases that provide you mappability between microRNAs and biological processes. That doesn't exist at any place. So we had to improvise a little bit in order to get to that answer. Okay, so use case 3, a little bit more complex, right? Now, what I wanted to do is we wanted to find all those genes that are associated with one disease, and we want to learn about their association with some other disease, right? So in order to ask this question, we chose 2 diseases, Crohn's disease, which is IBD kind of disease, and the effect that it has on neurological diseases. So we're talking about gut-brain axis here. And then we want to start with a list of genes that are interesting to us, and then we want to learn about how they're associated with Crohn's disease and their association with neurological diseases. So in order to ask this question, again, we started with one study where we found differentially expressed genes between control and Crohn's disease patients. And then we took those genes and then we started learning about those genes, and we wanted to know their association with Crohn's disease. So you can use a system like Open Targets that will give you information about Crohn's disease. So you can provide Crohn's disease as an input, and it gives you all the genes associated with Crohn's disease. Okay, and then you can ask the question, out of all those genes that are known that we extracted from Open Targets, how many of those are associated with 338 genes that we found using the analysis that we did? And then that's where the thought ends, right? So how do I find all the neurological disease related genes that are also associated with Crohn's disease? So, there is no way for us to move forward. So if I were to use DisGeNet, which is again one of the backend databases that Protein Miner uses, it sort of lets you do that, right? It tries to find all those genes that are associated with Crohn's disease of the ileum, and then you can take those and then try to find their association with the neurological diseases, which is one of the categories that we have here. Okay, but there is no filtering here, right? You can't filter. So I can't go here, filter this. And even if you had that option, this would be so easy. But there's no way to do that here. And then let's say I want to download this and do it offline, like not on the system, but on my computer. It downloads each protein as a tab, information from each protein as a tab. And then out of that, I have to filter for each protein, everything that has to do with nervous system disorders, and that'll give me an answer. So let's say I have 100 proteins, it'll generate 100 tabs there. It becomes a nightmare if you're not a data scientist, right? So, and again, we try to do this using Protein Miner. As you can see, I can take all the genes that are highly expressed, and then those associated with Crohn's disease of the ileum, and then it pretty much boils down to a few genes there. And out of those, I can focus on the neurological diseases that I'm interested in. We focused on Parkinson's disease, we focused on Alzheimer's, and multiple sclerosis and things of that nature. So it's easy to just go from a concept to something that provides you information and start asking more complex questions, as I can say. Okay, so those are all the use cases that I have. Now, there are a couple of thoughts that I want to finish up with. So one of the thoughts is that, some of the things that I just showed you, it's possible to do it using some of the commercial systems, right? And then we have to start thinking about scalability. Let's say I want to expand it to some database that I'm interested in. Let's say I want to expand it to a database where somebody has knocked off a gene, and then they figured out what other proteins are affected as a consequence. Let's say there are 10 or 15 proteins that are silenced as a consequence of knocking off a gene. You also have information corresponding to toxicity associated with those knocking off that gene. So I don't want to overlay that information on top of what I already have. So I can bring in that database, I can integrate with what I already have, and then I can do all of that. All of that is possible within a sprint or two. So we are talking about 2 weeks to a month where I can integrate that database. Number two, we might have some internal databases, data sets. Because of governance issues, we might not want to expose it to these commercial systems that we work with all the time. And because this is an internally developed system, such issues would not be a challenge. So we can rapidly bring in those data sets and integrate with all the public sources and try to provide the information. So with those two thoughts, I want to finish up my talk, and I'll take any questions that you might have.
Any questions for our speaker? All right, give him one last round of applause. Feel free to reach out to him after his presentation if you have any questions you're too shy to share with the group. I will hand it back over to you.
Okay, the next talk is by Alexander Sherman, who is a director, Center for Innovation and Bioinformatics from Mass Gen. Welcome.